Today we will continue our discussion about STK11 knockout impact. So in the previous video we have understand the migration assay that has been done in vitro or on the cells in the lab, on the pancreatic cancer cells within the lab. So in this video what we will discuss the experiment or in vivo experiment that is popularly known as CAM Xenograft Assay. So in this experiment what they have done is they have take the xenograft from an animal or the chicken and use it with this pancreatic cancer cells in which this STK11 is knocked out. So they did the experiment on TB30427 cells. So first for control they use the wild type TB30427. Next they use this another control, and the next two conditions are the different knockout clones of the cells that are developed as a single clone. That is STK11 knockout 1.5. That is this is the name that they have given, and STK11 knockout 2.2. And they use two different kind of dyes. First one is this HNE staining, where they can see that in the case of control the CAMs are shown in a transparent region and they seems to be intact in the control case. However, when you see the STK11, the CAMs are not that stable and are being infiltrated by the pancreatic cancer cells. These are the tumor masses. And same is the case with the other knockout. So in the case where STK11 is not knocked out, the rate is slower, whereas in the case where STK11 is knocked out, the tumor cells are spread faster. It can be seen here in this figure. Next one is cytokeratin. They use cytokeratins that is used to find out the epithelial cells migrations, and that is used to distinguish epithelial cells from the cancer infiltrated epithelial cells, normal epithelial cells with cancer infiltrated epithelial cells. So in the case of this TB32047 wild type and another control NC1.4, they do not see too much infiltration of this cytokeratins dye, or the cancerous epithelial cells is not spread too much. However, when they see it for the STK11 knockout cells, STK11 knockout samples, they can see that in both the cases the presence of cancerous epithelial cells are higher compared to the control case. And this panel D is the zoom in image of the panel B, where we can see that CAMs are here slightly transparent in region, whereas in the case of here the CAMs regions are seems to be slightly more, whereas in the case of this NC1 also we can see the transparent regions, whereas in the case of this knockout the transparent region seems to be fade out, and in the second knockout the impact is even higher than the first colony or the first knockout colonies. And in the zoom in view of this panel E and the zoom in view of the panel C, which is shown in the E, here also we can see that in the case of control, both the controls, the cancerous epithelial markers are not too much compared to the case of this knockout, where the cancerous epithelial markers seems to be much higher compared to the control case. So this indicates that from the in vivo studies also that STK11 knockout cancerous cells or pancreatic cancer cells which does not have STK11 knockout or which does not have STK11 will have more migration properties compared to the case of the pancreatic cancer cells that have STK11. So I hope from this in vivo experiments you might also get convinced to some extent that STK11 plays an important role towards this controlling of the migrations of the pancreatic cancer cells. If you need to understand more about their experiments and how they conducted their experiments, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.